Bioinformatics of shotgun metagenomics are complex, but generally, 3 steps exist: sequence assembling, gene prediction, and sequence similarity search. At each step, such as assembling and gene prediction, metagenomic sequencing data has unique features such as coverage of leads and frequency of cut codon can vary from contig to contig. So, dedicated software must be used compared to the single species genome assembling or single species gene prediction, because the metagenome data is a mix of many microbes. You need to use specific software for metagenomes. In the case of metagenomic analysis, not a amplicon sequencing analysis, in the case of shotgun metagenomic analysis, there are a variety of ways to estimate the taxonomic composition of a community, including using phylogenetic marker genes like 16 genes, or single copy genes like RNA polymerase beta subunit. The name is LupoB genes, or unique marker genes that genes are specific in specific taxa, or whole lead, or K-mar frequency. Since each method has its own merits and demerits, you need to select appropriate taxonomic assignment method depending on the number of samples and the purpose of the analysis. For functional composition inference of metagenomic data, many researchers use KEGG database to discuss metagenomic pathway abundance. For example, you can use Ghost Koala or Confirm Koala web service in KEGG for assigning a function in each metagenomic sequences. Although sequence similarity searches are often used for phylogenetic and functional assignment of large number of metagenomic sequences, BLAST, that is very popular tool, takes too much time. So, more faster tools such as Diamond or MM6 are often used. However, these tools are faster, but use a lot of memory, near the several 0.2 or 300 gigabyte. So you should be careful to use this software in your own computers.